Hello everybody, and good evening. Good morning to those of you in the US. I'm going to present to you today the status of the research at the Hebrew University during this past year, which is under the corona pandemic. I will give you a general picture of how research overall is doing under corona. I'll give you some numbers, but then I'll focus to show you what we were doing in respect with the corona research. Let's start with some numbers that can summarize the research or characterize the amount of research or success in research if you want. Here are the ISF, our main founder of the university, the Israel Science Foundation. And you see a growth in this past year from 293 to 321 submitted grants, and a comparable growth in the number of grants awarded. So this partially reflects our success and partially reflects the fact that we have younger faculty recruits in larger amounts, and we're growing, and therefore more success in grants. You can see that especially in equipment for new faculty, which grew tremendously from 7 last year to 21 this year. Several funding agencies gave funding directly for coronavirus, and we got a significant fraction of those, more than our fraction in the overall university horizon in Israel. We are still in a very good place in the European Union in the number of ERC grants that we are receiving. This is our position 10th in all of Europe, which is really impressive when some of these are not really universities, like Max Planck Institute is a huge entity. The Hebrew University is number 10 with 146 million euros obtained so far in the Horizon 2020 program, which is running for 5 years, and just 40 million euros this year, so more than the average amount that we used to get so far. So we're doing quite well. Here are some prominent grants, not related to Corona, but in the time of Corona, actually submitted before, but received during this time. All these amounts, more than $1 million per grants. These are impressive grants with people from the Faculty of Agriculture, the Faculty of Medicine, Life Sciences and Earth Sciences. The university is doing well on research. Isum, our tech transfer company, is also doing well. Many licenses, research agreements, spin-offs, and funds coming to the portfolio companies that we have. Our commercial activity is also going well. These numbers reflect more or less the averages that we have in good years. So this is doing well. Isum has not been hurt by Corona, which is also good. Again, through Isum, we also have several grants coming for research in Corona, in things which are practical, fighting the disease in several ways. Okay, so now let me get to the main focus of this talk, which is a little taste of our Corona research. And this comes in several avenues. We have prevention, different ways to prevent people from getting infected, vaccination and treatment of the disease, understanding the implication for society, the psychological implications on people, and the economical implications, and also a very strong effort in screening, understanding who is sick and who is not, so when we isolate people, we can do that differentially. Okay, so here is an example of a way to prevent Corona, or to prevent people from getting Corona. Special coatings. And this one uses copper, which is well known to be an antiviral agent, and it is doped with some other materials. And you can see that research we have done at the university shows that together with this doping, we get a huge reduction, several orders of magnitude, in the amount of viruses that are left alive after this coating, and there are several alternatives to that. There are several different groups working on similar things, and this kind of technology can be incorporated into masks, into clothing, into the alcohol gel and soaps, etc. So this is a very important avenue. In the geography department, people are investigating the ability of the coronavirus to spread by doing simulations of people going from one building to another for all different reasons, and how the pandemic spreads, what are the most important things to prevent, and therefore give advice to policymakers when they take their decisions. In the business department and economy, we're investigating how people make their decision under the stress of corona. Are they more susceptible to listen to advice that they are getting? Are they more risk averse when they are doing their financial decision? And how do they respond financially to the pandemics? This research already shows that there are important changes that people are making because they are afraid the future is not as clear as it used to be. Another interesting research by Shachar Arzi from the Faculty of Medicine is MRI images of the brain, where he monitors the functionality of social interactions and where they are located in the brain and what they are influenced by and what they are connected to. And that informs us about the ways to prevent the harmful effects of social distancing. There is even a product that he developed that allows people to choose better what are the way they want to spend their times and how, what kind of connections they want to keep with their friends and where to invest their time and energy. A very big push that we have at Hebrew University is screening and treatment of coronavirus, and I'll focus the rest of my talk about these 2 points. So let's start with screening. There is a big effort by Nomi Habib and Nir Friedman who came up with an ingenious method to detect the virus simultaneously in many people. So the way it works is they collect samples from several people that need to be detected. This can be 1,000 or maybe 10,000 people in the future. They do some sophisticated procedure to bind some marker to each one of these examinees. Then they mix the samples from everybody together and they throw everything into the sequencing machine. This is a very different kind of machine than the PCR machine that you usually use to do tests. This actually gives the DNA sequence of whatever you pour into it. So what this machine gives you is the DNA of the virus if it's there, but because it was bounded before with a marker, you can tell which of the examined people has the virus or contributed the virus to the sample. So when the virus comes to this machine, it could come from this guy and this guy together, but each virus will be connected to a different marker, and we will be able to tell that this guy gave the virus, this guy gave the virus, and those guys didn't. And in this way, they already succeeded to do 10,000 people in one shot, and they believe they can do 10,000 people later on. So what are the advantages and the disadvantages of this method? First, many people together, and therefore it's going to be much cheaper. Estimate is about $2 per test, which is maybe 10 times less expensive than what we currently have. It is a sensitive method, perhaps not as sensitive as the standard PCR, but still sensitive enough. So it's very good for screening of population which is not suspected to be sick. It's inexpensive, we already said, and the added values is that you get detailed data on the virus and on its mutation. You can actually say that the virus, the coronavirus, that this guy has and that this guy has, they're not exactly the same, and maybe you can figure out from that, for example, who contributed the virus to who, what was the chain in which the virus spread in the population. So very accurate information can come from that. So you can also use the same technology, or basically while you're doing the corona test, you can also see some other kind of viruses that will be attached to the same markers of the same people, and you can say which of them has a flu and should stay at home because he has a flu rather than because he has corona. Another effort on the way to find treatment and vaccination for the coronavirus is an effort at the Faculty of Medicine by Alex Rubinsky, Ilan Rosenshine, and others. And in this, they take different parts of the virus, and they see the reaction of the body, the antibodies produced in the body, to each of the parts separately. Most research in the world, this is done in many places in the world, but most regions in the world, they look at the spike, while the approach of this group was to take a whole holistic approach and inspect the response of our body to each different part in this virus. And each part like that activates the immune system is called an antigen, and they managed to build a large array of antigens located at different places of the coronavirus. It could be at the spike, but even different places at the spike, or at different places at all. And they produced a list of responses of different people to the different parts of the virus. Now, computationally, they take this and decide what are the best combinations of sites that are needed in order to attack the virus. And this is very important because when you give treatment to a specific virus, the virus usually mutates. And if you're only attacking a specific point at the virus, after a certain time, which depends on the kind of virus you're dealing with, you get an immunity of the virus to the treatment because the virus went through mutations. Their approach that looks at different parts of the virus will try to prevent that by attacking several different points simultaneously by the same treatment. Another very interesting effort led by Ofer Mendelbaum, again from the Faculty of Medicine, is to make the virus ineffective in the following way. The virus, through this spike, the same spike that we have seen before, attaches itself to a certain receptor at the body called ACE2. So it's this spike that is being attached to this receptor. So what can you do? You can avoid that by taking some particles which are very similar or actually identical to the receptor in the body and spread them in the patient. Those receptors will go and block the binding points of the virus and make this virus ineffective. It won't be able to attack the body anymore because all its binding points will be blocked. Similarly, you can take that part of the virus, distribute it in the body, and that will go and block the receptor at the body and will not allow it to accept the virus. So what these figures are showing here and here is that actual experiments on mice already done show that you can decrease the amount of mortality that you get in mice from coronavirus by a significant factor if you do this treatment. So this seems like a very promising avenue. And of course, we should now test it on humans to see if it works in real life. To do all that, we need special laboratories that can host these experiments under safe conditions, safe for the environment and safe for the researchers that are getting into the laboratories. The level of safety that we need is BSL 3. Almost the highest level exists. So we need something with hoods that take the air outside, completely sealed from the surrounding, very good ventilation, and a significant protection for the people working in there. We have already an operational lab like that. This is an old lab that we refurbished and brought it back to functionality in order to deal with the coronavirus. And this is actual team that is dealing with some experiments right there. And there are already external companies that are asking us to work in this lab because this is the only lab like that in Israel that allows researchers to use it and do experiments in it. This lab is just a small version of what we're going to have at the end. It's already operational. This is what we're aiming for, a bigger room which also has option to treat animals inside. This is our design, which is over 100 square meters. labs, and again, it's going to be a national lab serving all the universities in Israel and other companies. It's important to have mice in there, I already said, and we need also to produce these mice. There is a problem that mice inherently are not susceptible to coronavirus. They don't have the same receptor ACE2 that we talked about before that can absorb the coronavirus and allow it to infiltrate the cells. So mice are basically immune to corona, and this is good for them, but it's bad for us because we can't test corona on mice. So there is a way to make a modification to the mice to be able to make them susceptible for corona, and then you can make experiment on them. There are several different ways to do that. One is to infect them with some other virus which changes their cells and make them now susceptible to corona, or completely change their genome in our GEM facility and create a transgenic mice. We're working on both options. The first option with the lentivirus is already working, and we have mice that are susceptible to corona produced in this way. The second option we tried. The construct that you need to inject into the cell is too long for the standard method to work, so that first tried failed, and we're now trying in a second technique to try to make transgenic mice this way. Meanwhile, we're buying mice from external companies that already managed to do transgenic mice, and we're ordering some other mice from China. So we're trying basically to get this set up both internally and from external sources. So let me summarize here our different efforts. We have efforts in treating the disease. We have the production of antigens and therefore antibodies that are fighting corona. We are on our way, so haven't done that yet, to inject them into animals, and therefore those animals will produce the antibodies that can already serve as a passive vaccination for this disease. We found agents that can block the attaching point of the coronavirus to the cells. This is done completely on animals, and we should do that in larger numbers and larger quantities to improve the result and to test it on humans. We're working on producing animals. Some of them we already got. We're working on producing our own, some of it successfully, some of it not so far. And the lab is expected to arrive here in December. So I hope that by December we can have the entirety of this operation working and with good progress on corona. So I hope you got a chance to see just a glimpse of the vast amount of activity that is going on at the Hebrew University, the breakthroughs that we achieved so far, and our plans for the future, which I really hope we can tell you more about next time. Thank you very much.